Good morning, and thanks for joining me. I'm Rachel Dunn. I'm the Director of the Division of Pharmaceutical Analysis in the Office of Testing and Research, which is within the Office of Pharmaceutical Quality at CDER. And today I'm going to cover some examples of how our lab generates data to support the assessment of generic complex drugs. I'll start by describing the Office of Testing and Research's role in generic drug science and how our laboratory-based science and research programs support the evaluation of generic drugs. I'll provide a couple of examples of ongoing research projects at OTR and that address regulatory issues with complex products, including peptide therapeutics and inhalation drug products. But first, what is pharmaceutical quality? A quality product of any kind consistently meets the expectations of the user. Drugs are no different. Patients expect safe and effective medicine with every dose they take. Pharmaceutical quality is assuring every dose is safe and effective, free of contamination and defects. It's what gives patients confidence in their next dose of medicine. The Office of Testing and Research has 3 divisions. 2 of them are located in St. Louis, including my division, so I'm coming to you from St. Louis, Missouri this morning. And our 3rd division is located in White Oak, Maryland. Our laboratories have a ton of analytical instrumentation to support our testing and research programs. A few of our labs and some of our instrumentation are shown here, but this really only represents a small portion of the instrumental capabilities we have in the OTR labs. So what do we use all this state-of-the-art instrumentation in labs for? OTR's lab science supports the OPQ mission to assure quality medicines are made available to the American public, and we do this in 4 main ways. We develop analytical methods and generate data to support guidance development, policy making, and science-based responses to industry and the public. So we aim to address immediate regulatory needs and anticipate future regulatory questions through our laboratory research. We do a lot of product testing in the laboratory to support pharmaceutical quality surveillance and compliance actions. OTR scientists conduct research to evaluate and verify information in applications for drug assessment and approval, so we consult on specific scientific issues for regulatory submissions. And finally, OTR uses its laboratory-based research and testing capabilities to support the FDA's public health priorities, so we can better respond to rapidly evolving public health emergencies. So our research is focused in 5 main areas, which I'm showing here, but specifically today, we're talking about OTR's lab work that supports the review and assessment of complex products. Complex drug substances and products require good analytics for good characterization. We're working to develop advanced analytical methods and strategies for the characterization and comparative analysis of complex drug substances. And we're also working to develop in vitro approaches for characterizing complex formulations and dosage forms to assist in product development and in regulatory evaluation. So let's go ahead and talk through a couple of examples of OTR's lab research in support of these priorities. First, I'll introduce using advanced analytics for the characterization of generic peptide drug substances. Complex molecules like peptides, proteins, and biosimilars represent a large and rapidly growing drug market. Patents for many peptide-related RLDs are expiring in the next few years, and the number of peptide-related ANDAs submitted for approval based on their sameness to RLDs is dramatically increasing. Therapeutic peptides can be produced by a couple of different methods, and the method of production has an effect on the impurity profile of the drug. It's important for us to understand the impurity profiles so we can evaluate the associated risks of those impurities. Some QC methods may not be adequate for identifying and quantifying all impurities in a product. For example, HPLC with UV detection is often used for quality control, But if the chromatography doesn't allow for baseline resolution between all components, or if some components don't have a UV chromophore, HPLC with UV detection is really insufficient for demonstrating the quality of that peptide drug product. So in this area of therapeutic peptides, there is a real need for advanced analytics. OTR has a number of ongoing research projects exploring the use of liquid chromatography with high-resolution mass spectrometry, or LC-HRMS, in the characterization and impurity profiling of therapeutic peptides. So it's been used for many years in research, and it's been widely used by bioanalysis groups. And it has a lot of advantages. It has very high selectivity and high sensitivity. It has the ability to provide qualitative and quantitative data in a single experiment, and it provides very accurate mass determinations. But there are some real challenges associated with the technique also. Scientists need to be very highly trained to develop and use LC-HRMS methods. The resultant spectra can be very complicated, and the instruments themselves are very expensive. So OTR developed and validated an LC-HRMS method for 3 peptide drugs, and we demonstrated the suitability of this analytical technique and approach for the quality control of peptide drug products. So in this example here, you can see the total ion chromatogram of the peptide drug calcitonin salmon, and you can see the main peak for it, and then in the expansion down on the lower left, again, you can see the main peak and a number of peptide-related impurities that we were able to identify and quantify using this method. And we were able to identify and quantify several more impurities than were identified by HPLC UV, which is the USP monograph test procedure for assay and impurity of calcitonin salmon. So this demonstrated that this is a promising approach for quality control, and it also provided some guidelines to industry for establishing method validation and performing comparative analysis using LC-HRMS. OTR's research also aims to build the capacity for comparing impurity profiles of peptide drugs of varying origins. So as an example, we explored the drug impurity profile of teriparatide, which is a 34 amino acid drug used for treating osteoporosis. RLD was approved in 2002, and it's produced by recombinant expression in E. coli. The generic drug product applicants are producing their teriparatide by chemical synthesis. So if impurities are present at different levels in the RLD and the generic, that can cause problematic outcomes in patients. So it's more than just an academic exercise for us to dig down as deep as we can to identify and quantify peptide-related impurities in these drugs. It's really important because we need to identify the impurities and provide these data so that our colleagues in the agency can make risk-based assessments about the immunogenicity and the safety of these drugs. And the data that we collect in the laboratory to compare impurity profiles will also assist in regulatory decision-making. So here's the amino acid structure of teriparatide. Impurity profiles are going to vary based on the manufacturing process for therapeutic peptides, and I've listed some potential peptide-related impurities that we might find in these products. For teriparatide specifically, we expected potential degradation impurities to include the oxidation of methionine at positions 8 and 18 to methionine sulfoxide, and the possibility of the formation of a succinimide at position 30. And so those are highlighted in this structure. OTR scientists have been working on developing and optimizing an LC-HRMS method for teriparatide over the last couple of years. And we've developed a data-dependent workflow to identify and quantify peptide-related impurities in teriparatide. So I'm showing this here, and with this approach that I'm showing in this workflow, we were able to rapidly identify peptide-related impurities in several lots of the recombinant teriparatide RLD and impurities in synthetic teriparatides, which weren't present in the RLD. And we were able to do all of this with a mostly automated data analysis. So this workflow that we've developed, and I'm showing here, can be used to characterize impurity profiles of other therapeutic peptide products. And we have some ongoing research in OTR labs doing just that. So OTR scientists are producing data to develop regulatory guidelines that will support industry for developing new products, and also will support regulatory decision-making by the FDA. And now we're going to change gears, and I'll discuss another area of focus for research on complex drugs in OTR, inhalation products. So metered dose inhalers, dry powder inhalers, soft mist inhalers, these all include a drug and a device. And these products present complex interactions between formulations and device design characteristics. The evaluation of bioequivalence for complex generic inhalation products can be difficult due to these complex drug-device interactions. The agency recommends a weight of evidence approach for demonstrating bioequivalence of generic inhalation products, but this approach can be time-consuming and very costly. So OTR scientists are doing some research to explore alternative and predictive in vitro methods to make bioequivalence demonstration more cost-effective and more time-effective. And we've investigated some biorelevant mouth-throat models for the evaluation of the aerodynamic particle size distribution. And then we also were looking at alternative methods for measuring particle size distribution. So current in vitro methods and equipment for measuring aerodynamic particle size are designed for quality control, and they have limited predictability for drug deposition in vivo because they don't adequately mimic airway geometry and actual use conditions. So you can see the USP mouth-throat model here. It's a straight tube with a 90-degree bend. It doesn't look a lot like a human mouth or throat. But several biorelevant mouth-throat models have been explored by academia and industry. These models vary in size, internal volume, geometry, the materials they're made out of, and they've been created with the intent of providing better in vitro, in vivo correlation. So in OTR, we've done a study to compare a solution-based metered dose inhaler product to a suspension-based metered dose inhaler product, along with 6 different mouth-throat models, or using 6 different mouth-throat models. The study that I'm showing here shows the biorelevant models, so the 4 on the right-hand side of this bar graph, provided a better prediction of mouth-throat deposition than the USP model did. So the in vitro results represented by the bars more closely match the in vivo results represented by the dotted lines when the biorelevant mouth-throat model is used. These results are product specific, but choosing a suitable biorelevant mouth-throat model for evaluating a drug product may provide better in vitro, in vivo correlation to assist in product development of inhalation drugs. We've also investigated the use of morphology-directed Raman spectroscopy as an alternative approach to the comparative clinical endpoint bioequivalence study. So MDRS is a combination of an optical microscope, which allows you to do particle sizing and morphology analysis, and a point-and-shoot Raman spectroscopy for chemical identification. It's especially useful in products where the drug and the excipient are both in suspension, and you need to be able to differentiate between components so you can measure the particle size distribution of the API independently from the other components in the product. And this is really difficult to do using conventional methods, but MDRS is very well suited for this. So in OTR, we've been engaging in research using MDRS to provide data for new guidances that will aid in the development, optimization, and validation of MDRS methods. There are really some great advantages of this technique. It's non-destructive. You can chemically identify different components of a mixture, and selectively measure the particle size distribution of a component. However, it's limited in that you can't measure particle sizes less than a micron, so you'd have to use an orthogonal technique for submicron particles. The research that we've done on MDRS as a technique will be used for the development of a regulatory guidance outlining what needs to be considered when developing and validating a new MDRS method. We are evaluating alternative in vitro techniques for evaluating new types of inhalation devices. So the first soft mist inhaler was approved in 2014, and the first nasal powder inhaler was approved in 2016. These devices offer new aerosolization methods for the drug products. We're evaluating laser diffraction as an alternative in vitro method to measure the particle size distribution of these newer types of aerosolization devices. There are some great benefits to laser diffraction. It allows for very rapid particle size distribution measurements on the order of seconds. And it also allows for really versatile sampling of products. You can sample suspensions, emulsions, dry powders, aerosols. Laser diffraction has a few limitations. I mean, it can't chemically identify the components. And there are also some potential influences on the measurements depending on the conditions. So in OTR, we're evaluating laser diffraction for particle size measurements, and we're varying the conditions under which we make the particle size distribution measurements. So we're varying temperature and humidity. We're using co-flow or not, and we're varying the distance between the inhaler nozzle and the laser. So the FDA is going to use the data that we are generating to develop new guidance documents and provide recommendations for this analytical method. So in summary, OTR's science and research work supports the Office of Pharmaceutical Quality's mission to assure quality medicines are available to the American public. And generic drug research is an integral part of OTR's work. Our scientists conduct research and we generate data to support guidance development and policy making and to anticipate regulatory needs. We do collaborative laboratory-based research to answer questions and to evaluate information in generic drug applications. And the data that we generate in the lab can allow for risk-based assessments of new drugs, including potential for immunogenicity. And now for the challenge questions. Advantages of LC-HRMS for the characterization of peptides include A, qualitative and quantitative data in a single experiment with no need for a UV chromophore and no specialized training needed for QC lab staff. B, qualitative and quantitative data in a single experiment. It can resolve co-eluting analytes and there's no specialized training required for QC staff. C, qualitative and quantitative data in a single experiment. It can resolve co-eluting analytes and there's no need for a UV chromophore. Or D, there's no need for a UV chromophore, there's no specialized training required for QC staff, and it can resolve co-eluting analytes. And the answer is C. Challenge question number 2. A particle sizing technique that can also differentiate between chemical components is A, morphology-directed Raman spectroscopy, B, laser diffraction, C, biorelevant mouth-throat models and cascade impactor analysis, or D, dynamic light scattering. And the answer is A, morphology-directed Raman spectroscopy. Thank you very much for joining me today and for your kind attention. I hope you learned about some of the ways that OTR's lab-based research programs support the FDA's mission and the generic drug industry. Thank you.